Hi guys, we are going to see about physiology of white blood cells in detail. At the end of this video we will know about introduction, classification, morphology, normal value, lifespan, properties, function, variations. A leukopoiesis is called development of WBC. Introduction. White blood cells or leukocytes are the colorless and duclated elements of blood. Compared to RBC, WBC are larger in size and lesser in number. WBC play very important role in the defense mechanism of body, by acting like soldiers and protecting the body from invading organisms. Classification. They are divided into two groups depending upon the presence or absence of granules in the cytoplasm. Gaulocytes with granules. Agaulocytes without granules. Gaulocytes. Depending upon the staining property of granules, they are divided into three types. Neutrophils granules take both acidic and basic stains. EOSINOPHILS granules take acidic stains. BASOPHILS granules take basic stains. Agaulocytes. They have plain cytoplasm without granules. They are monocytes, lymphocytes. Morphology of white blood cells. Neutrophils. Diameter 10 to 12 micrometer. Nucleus. They have multi-lobed nucleus. And they also called polymorphonuclear leukocytes. The number of lobes varies from 1 to 6. Cytoplasm. The granules are fine and small in size. The granules appear violet in color. When stained with Leishman's stain, the granules take both stains equally. Neutrophils are amboid and phagocytic in nature. EOSINOPHILS. Diameter varies from 10 to 14 micrometer. Cytoplasm have larger granules, which stain pink or red with eosin. Nucleus bilobed and spectacle shaped. Rarely trilobed nucleus present. Basophils. Diameter 8 to 10 micrometer. Cytoplasm coarse granules. They are stained with purple blue with methyl blue. Nucleus bilobed. Monocytes. They are larger in size. Diameter 14 to 18 micrometer. Cytoplasm clear without granules and large amount of cytoplasm. Nucleus round, oval, horseshoe shape, bean shape or kidney shape. It is placed in the center of the cell or pushed to the one side. Lymphocyte. Cytoplasm don't have granules. Nucleus oval, bean or kidney shaped. They occupies the whole cytoplasm. A rim of cytoplasm may or may not be seen, depending on the size. They are divided into two types. Large lymphocytes, younger cells with a diameter of 10 to 12 micrometer. Small lymphocyte, older cells with a diameter of 7 to 10 micrometer. Depending upon the function they are divided into two type. T lymphocyte, concerned with cellular immunity. B lymphocyte, concerned with humoral immunity. Normal value. Normal WBC count ranges between 4,000 to 11,000 per cubic millimeters of blood. Differential WBC count. Type of WBC, percentage, absolute value per cubic millimeter. Neutrophils 50 to 70%, 3,000 to 6,000 cubic millimeters. EOSINOPHILS, 2 to 4%. 150 to 450 cubic millimeters. Basophils, 0 to 1%, 0 to 100 cubic millimeters. Monocytes, 2 to 6%, 200 to 600 cubic millimeters. Lymphocytes, 20 to 30%, 1500 to 2700 cubic millimeters. Lifespan of white blood cells. Neutrophils, 2 to 5 days. Eosinophils, 7 to 12 days. Basophils, 12 to 15 days. Monocytes, 2 to 5 days. Lymphocytes, half to 1 day. Properties of WBC. Diapedesis, it is the process by which the WBC squeeze through the narrow blood vessels. Amboid movement, neutrophils, monocyte and lymphocytes show amoebic movement characterized by protrusion of the cytoplasm and change in shape. Chemotaxis is the attraction of WBC towards the injured tissue by the chemical substance at the site of injury. Phagocytosis, neutrophils and monocytes engulf the foreign bodies by means of phagocytosis. Functions of WBCs. Neutrophils. Neutrophils provide the first-line defense mechanism of the body. They wander freely all over the body through the tissue. Mechanism of action, they released in large number from blood. They are also produced from progenitor cells. They are moved by diapedesis towards the site of infection by means of chemotaxis. Chemotaxis occur due to the attraction by some chemical substance called chemoattractants which are released from the infected area. If the chemical substance increase, the neutrophil get firmly attaches to the site of infection. Each neutrophil can hold 15 to 20 microorganisms at a time. First, neutrophils engulf the bacteria and destroyed by phagocytosis. Eosinophils. It provide the defense against parasitic infection and allergic condition like asthma. It is responsible for detoxification, disintegration and removal of foreign proteins. Mechanism of action. They attack the microbes by secreting some special type of cytotoxic substance. These substance becomes lethal and destroy the parasites. They are eosinophil peroxidase, major basic protease, MBP, eosinophil cationic protein, ECP, eosinophil-derived urotase, interleukin 4 and interleukin 5. Basophils. It play an important role healing and acute hypersensitivity reaction. Mechanism of action. It execute the function by releasing some important substance from their granules such as heparin, which is essential to prevent the intervascular blood clotting. Histamine, bradykinin and serotonin, which produce the acute hypersensitivity reaction by causing vascular and tissue response. Protease and myeloperoxidase, that exaggerate the inflammatory responses. Interleukin 4, accelerate inflammatory responses and kill the invading organisms. Mast cell. It is a large tissue cell resembling the basophil. Usually, they are found in the blood vessels and they don't enter into the bloodstream. They are predominantly seen in the skin, mucosa of the lungs and digestive tract, mouth, conjunctiva and nose. Functions. They functions along with basophils and produce hypersensitivity reaction like allergy and anaphylaxis. The cell-secreting substance like histamine, heparin, serotonin, hydrolytic enzymes, proteoglycans, chondroitin sulfate, arachidonic acid derivatives such as leukotriene, LTC, and prostaglandin. Monocytes. Largest cell among the WBC. They are phagocytic nature, motile. They provide first-line defense along with neutrophil. It is the precursor of tissue macrophage. Mature macrophage stays in the blood for few hours afterwards it enters into the blood and becomes a tissue macrophage. Example, Kupffer cell in the liver, alveolar macrophage, macrophage in spleen. they secreting certain substance like interleukin-1, IL-1, colony-stimulating factor, MCSF, and platelet-activating factor. Lymphocytes. They are classified into two types. They are T lymphocyte, B lymphocyte. T lymphocyte are produced from bone marrow and stored in lymphoid tissue of lymph nodes, spleen, bone marrow and GI tract. B lymphocyte are discovered in the bursa of Fabricius in birds, situated in the cloaca of birds. The bursa is absent in mammals and the processing of B lymphocyte take place in bone marrow and liver. Stored in lymphoid tissue of lymph nodes, spleen, bone marrow and GI tract. Leukopoiesis. The committed pluripotent stem cell give rise to WBC through various stages. Factors necessary for leukopoiesis. Factors necessary for leukopoiesis. Leukopoiesis is influenced by hematopoietic growth factor and colony-stimulating factor. Colony-stimulating factor, they are proteins which cause the formation colony-forming blastocysts. They are three types. Graulocyte CSF, GCSF, Graulocyte monocyte CSF, Gram CSF, monocyte CSF, MCSF. Hematopoietic growth factors are. They are interleukins 3, 6 and 11 and stem cell factor, steal factor. Generally these factors induce the proliferation of PHSCs, uncommitted pluripotent hematopoietic stem cells. Thanks for watching. DENTOLOGY notes. Please subscribe.